you very much for Dr. Fan for the nice organization of this great event, putting many old friends and also wonderful researchers together. And thank you very much, Mike, for joining us. I think it's a very good time to talk about this paper, which has been published last month. So I think it would be a better time to share some of my views on bio-robotics and biomimetics. So today I will specifically talk about aerial-aquatic robots capable of crossing the air-water boundary and also hitchhiking on surfaces. These are two different capabilities, but they are inspired by Remoras. So let's talk about Remoras. Before I talk about Remoras, I want to talk about its host. It's a shark. In 2011, when I was at Harvard University as a postdoc with Professor George Lauder, I worked on shark skin, which is a very unique structure. If you can look at this video, there are thousands of shark denticles on just a 1 millimeter square meter. So it's very small, and we identify its structure, reconstructing a 3D model, and then build the first multi-material 3D printed shark skin. And then we published a paper on JEB, featured as a cover. What's interesting is how getting involved or engaged in the Remoras research is because of this picture. That's why I mentioned shark skin, because I want to find a very good picture of the shark. So that's this one. Although I did not eventually picking this one on the right, if you see my cursor, but when I search online, search on Google, I search sharks, and then it comes out a lot of lovely pictures. But many of the pictures I see, there are some mysterious hitchhikers on the shark bodies. If you see the red dashed box indicating right here, I was curious, what is that? And very fortunately, there are many excellent researchers who knows biology. And then I talked to some of my colleagues, and then he took me to the Harvard Natural History Museums. So I think it's still there, but a lot of people visited it upstairs. You can see a lot of public exhibitions. You can see dinosaurs, you can see monkeys or birds or fishes, a lot of things. But there's a negative or minus levels. In -1 levels, there's a fish connection in the natural history museums. We call this fish connection dictionary, where there are stores 1 million fish species and putting every fish species in the jars. So people preserve this well. And I would like to thank this person, Carson Harvey. He's from the Harvard Fish Connection, working for over 40 years. And he connected fish around the world. And I asked him, okay, can you get me a remora? I want you to take a look at what remora looks like. And Carson is very nice. He showed me to the jars of the remoras. So that is my first sight, or my first view to the remoras. So you can see my hand, that's my hand here. And this remora is here. It's a remora specimen. But if you look carefully at the label down there, it's September 20th in 2001, which means that this fish was caught over 100 years ago. It's still there, but it's very useful. You know, it's my first views on the remoras, and then I took the remoras out and look at how it looks like, and then dissecting it. So I think that's my first impression of these animals. I also look online and see is there any literatures about remoras, but unfortunately I did not find any papers about remoras. So I think we need to do some work on this. Here is a video from National Geographic, which shows a remora can hitchhiking to a lot of large marine animals, including the sharks, the whales, and also the human divers and sea turtles, and also the oxygen pumps, many kind of different types of surfaces. This is because it has a very unique disc pad for adhesion, so which is located on its dorsal side of its body. So this adhesive disc, they can generating enormous amount of force, including the pull direction and also the shear direction. So to investigating what is the mechanism of the adhesive disc, we need to do some research work on the specimen and also on the live Remoras. But let's talk. If we look at the previous adhesive mechanism in nature, we can see that a lot of people study geckos because the geckos using variable forces for dry adhesion, they can climb on the smooth surface, on the glass, along the vertical wall. And also people study the tree frogs whose the mechanism can be attributed to wet adhesion, where capillary effects playing the major functional roles. But until 2013, when I started the Remora research, not too many papers characterize the underwater adhesion. So Remora is a very good biological examples to get into the underwater adhesion mechanisms. So that's why we choose this animal to do further research. We get a Remora specimen from the jars and cut its head and do the CT scan. So the CT scan is the normal scanner that people scan in like the X-rays, which allows you to see the bone and any bony structures. But a special CT scan allows to see both the muscles, which are soft tissues, and the bony structures. That's what this video shows. We can see that the white color indicates a bony structure, while the red color indicates a flesh. So this video show provides a very direct information that Remora disc has both rigid tissues and soft tissues. So we reconstructed a 3D model, and we can see that its basic morphological feature is it have rows of, we call this lamellae. You can see there are many rows, and on each rows there are several sub-architectural structures. If we zoom in using scanning electron microscope, we can see that there are 3 or 4 rows of spines. So these little tiny spines have 200 micrometer in bottom diameter and around 500 micrometer in its height. And more specifically, the lamellae we call, there is a ventral process right here. It's not a static structure, unlike the geckos. The gecko also have a lamellae, but it cannot move. The Remora have the lamellae, and it can be directed or moved by the depressor muscles and the erector muscles. If we using a simple arrow to describe its motions, it can erect up or float down by the muscles. So it's dynamic. These basic features can be described using a biomimetic mechanism. This is a very simple because a lot of people here, like Professor Hui-Chan Zhao, study soft robots. We can use a very classic, like a soft actuator actuated by pressure. They can pressurize or depressurize and generating the linear translation. And then this linear translation can rotate in the ventral process and eventually induce an MLA pitch motion, just like the biological counterparts. So this is the how one can do the biomimetic model of the lamellae of Remoras. But to understand the entire mechanism of the disc, we need to fabricate a whole disc. So at that time, to fabricating such kind of hybrid or structure combined both soft materials and rigid materials, its technology is still lacking. So we use the a multi-material 3D printer, where you can see that different color represents different stiffness. The blue represents rigid, and the red represents soft. So you can see this is how we design the whole disk. And we are using the printer to print the whole disk out. There's one structure whose material stiffness is beyond the capacity of the 3D printer. It's the spinels. So we use carbon fiber plates and cut it by the lasers. And we can use this one technologies to highly mimicking the structure of the spinels. And Kevin Chen from MIT helped me to implement these tiny structures. After cutting it out, we have many rows of the spinels. It's made of carbon fibers. We insert the rows of enamel into the 3D printed disk, and we flip it back and adding the soft actuator to play the function roles of the muscles. And then we get the disk. So on the right, you can see the videos. The enamel is moving, just like the Remora. It can do multi-degrees of freedom motions, like they can rotating, follow a sequence. So here is a side view of how the muscles move the enamel up and down, just like the biological counterparts. On the left, this microscopic images shows spinels inserted into the soft material of the disk and then extruding out with a certain angle, which angle is biologically relevant to the animals. And if we compare the bio-inspired disk with a live Remora disk, its motions, we can see that it's very similar and biologically relevant regarding both directing up and fold down motions. And also, we measure the force. I skip a lot of details, but I'm going to tell you the major results and conclusion of that. So, and the disk prototype can generating a pull force along the normal direction up to 340 times of its own weight. So if you give a absolute value is over 100 newtons, or I will not skip some of the details, is the pull force along the normal direction can reach up to 340 times of its own weight. And also the shear weight, and also the shear force along the frictional direction, they can be tuned by moving the enamel up and down. So you can see that. So this provides deliver a very direct information that the Remora disk, the frictional force can be tuned by its motion of its inertia enamel. So to demonstrating its robotic applications, we implement the disk to a very simple robot. It's called ROV with cables and battery outside. It can swim to a surface like a Remora and will also detach. But there are two major problems for this prototype, for this robotic prototype. Number one, it have cables, so it cannot move around with a large working area. And number two, its a disk cannot be fitted or adaptively adhesion to all sorts of surface, including the curved surface or very rough surface or biofouling surfaces. So to overcome these problems, we hope that this disk can work not only on the water, but also in the air. So let's go back to look at the Remora, how they attach to the live animals. So this video made by Megan Cannawell from Science. So she showed that this is a dolphin leap out of water for fun, and when they spinning in the air trying to dislodge the Remoras, you can see that the Remoras can firmly attach to the surfaces of Remora, of the dolphins. Is there any problems? And also, if you look at the morphological feature of the biological Remoras, they can stick to surfaces with many holes, and just by even part of the disk. So if you can see here, this is indicated by the dashed lines, a partial disk attachment that can still remain firmly and robust attachments. And also on the right image, you can see that the Remora can just using just one half of its disk to attach to a glass substrate. How this happens? We're using a very special technology called FTIR, frustrated internal reflection. So by shedding the lights from the side of the glass, we can see that how the disk was attached to the surface, and the shining parts are the area that contact the surfaces, while the black area indicating the cavity or the chambers formed by the Remora disk. So in total, there are 25 rows of the chambers or cavities being formed. So to understand how the Remoras can generating the redundant adhesion, we put the disk under the microscope. We see that there are connecting tissues between the rigid lamellae and the disk. Let me play this video again. You can see it's right here. That's a connecting tissue. So we mimic these features and incorporating that into the robot. But before we start a complete model, we also dissecting the Remora fish, and we see that this is a cross-sectional view of the Remora. So here is the lamellae body structure. Here is the bone lamellae softer tissue, and right here the yellow is Remora. So can you hear me?
Yes, we can.
Okay, yeah, I just repeatedly check. Sorry for that. So here's the connecting tissue with the disk lip. So this is the first prototype that form redundant adhesion with many lamellae compartments. And also by applying the redundant adhesion, we can use part of the disk to attach to the surface and grasping many objects. For example, they can grasp in like objects with a curved surface, and also tape like this, and also concave surface objects, and also like the shell or conch. And to further enhance the frictional force, we found that the Remoras actually have the vents and vessels to hydrostatically enhance its adhesion. So we separating this contact stage into pre-contact, the redundant adhesion, and finally the frictional enhancement. And on the right video shows how we implemented this. The basic idea of this one is to using water pumps right here to direct the liquid directions, so they can pour the water in to fall down to erect up or fall down the lamellae to change its frictional force. So if you look at the video, so this is its mechanism of how the hydrostatic enhanced adhesion was generated. The water pumps goes down and then erect the lamellae and change the pressure inside and outside of the disk. And so here's the comparison of the 2017 paper version and 2022 version. You can see that 2022, even part of the disk, its adhesion was break, but still maintain robust adhesion. So let's put everything together. We're using two very small hydraulic water pumps and using multiple layers to putting all the functional layers of the disk together. We implemented untethered, hydrostatically actuated Remora disk, so that with this device, they can be mounted or assembled to many robots without any tethers. And also we tested its performance. We can see that the water pumps can erect the lamellae and also can detach the disk by using small motors right here by using cables. So now we want to make the disk hitchhiking not only on water, but also in the air. So that's why we need to consider aerial-aquatic robots that can make faster transition, swim on the water, and fly in the air. So if we look at the previous work on aerial-aquatic robots, the flapping wing aerial-aquatic robots by Kevin Chen, which took 129 seconds. And this work is by using fixed wind, but by combustions by ejecting water out, which took over 800 seconds by Mirko Kovac. And also this one fixed wind took, it's a transition period took 1.85 seconds, but it's very hard to control. While some other people tried the biomimetic way such as the squid, to mimicking its function to jump out of water, which took around 4 seconds. And also people use cod rotors to make the transition, but it requires many rotors here, which took around 1.5 seconds. But the problem for this prototype is it still have cables. The battery is outside of the robot. So the question now is how we can make our untethered rapid transition robot that can fly in the air, swim on the water, and also make the smooth transition. So that's our question. So here is a first solution is we're using a self-folding propeller to speed up the transition. If you look at the video, we can see that when it's rotating on the water, its rotors self-folded. And when it touches the water surface, it's self-spanning. So the rotor spans out. So just like normal aerial foils. So by applying this approach, the transition speed was significantly enhanced. If you can see the video on the right, so on the from the underwater to the air, it only took around 0.5, less than half a second. And also it can reversibly going up and down or going inside and out of the water. And we also compared the robot transition speeds versus time using different propellers. So let me check again. Can you hear me well now?
Yes.
Okay.
I think it's perfect.
I'll keep on. Yeah, sorry for that.
No problem.
So the robot transition speeds versus time using different propellers. The first row is the self-folding propeller we used in this paper, which can see that from water, there's a transition period, and into the air, the transition stage took around 0.39 seconds. So, but the commercial ones, the transition took 1.06. And it's exactly the same stroke out, which means the power inputs or the rotor speeds. In comparison, the fixed unfolded, that's exactly the same prototype, but its blades was fixed at unfolded state. It takes around 1.4 seconds around that. So the results show that by applying the self-folding propellers, its transition speed can be much better than the other cases. So we have done a lot of experiments on this. This is just a representative data we took from the paper. So after solving the transition problems, it can swim on water and fly in the air. We want to test, is the robots capable of hitchhiking on moving objects, especially when attached to a surface whose speed is faster than the robot itself. Can they do that? So we took the high-speed videos of how they attach to the moving surface and also measure the force. So on the right panel, figure panel, you can see that we divided the process into 3 stages. The stage 1 is approach, okay? So, and the stage 2 is contact. During the contact stage, there's small time frames. You can see that it's sliding backwards. and eventually reach equivalent states and get into catch the following stage. And during this, we can see how the force changes during this hitchhiking or moving surfaces process. And eventually, we also want to test it. Is there robots can do something like grasping objects out of water? So we first look at some other animals like the kingfisher. They quickly dive into the water and then catch its prey like a fish and fly away. So we hope to do exactly the same thing. Like the robot can fly in the air and also quickly dive into the water and then pick it out. So that's our demonstration shown in this high-speed video, which has been significantly slowed down, which allows you to see the whole process. And all these previous slides are laboratory experiments. We hope to show you how the robot's performance in the wild. So we took the robots to the mountain stream, and where we can see that the robot can fly out of the mountain streams within half a second, and also they can firmly attach to the slippery surface of the rocks with very stable views of the camera on board. And here is the students, my student Lilei is trying to attach to a very slippery and uneven surface like the rocks. So this is a freshwater experiment. We also tested this experiments in seawater. So this is in the ocean depths where the robot can fly out of water, shoot itself out of water within half a second. And also this robot can attach to a swimming robot and dive with a robot to depths up to like 10 meters and also record the information from the seabed as shown here. And also its depths can reach to around 10 meters. I think we're working on a robot that can reach a deeper depth. And also finally, we show that they can grasping a sample plate out of water, as shown here. With this, I think that the future perspective of this robot is we can use this robot for aerial observations and also make the transition, do the submerged structure inspections, or do the marine life survey or iceberg detection during the global warming, and eventually grasping some environmental sampling objects. We hope the robots can do that. But this is a perspective, a lot of work remains to be done. So before I finish up, I would like to introduce some ongoing Remora work because we are not satisfying with current progress regarding we have not understand enough about Remoras itself. Because according to this video, you can see that Remora actually very flexible, not only its body, but also its head and neck. So they can make a safe start with the yaw motions. They can quickly pitch up itself and make detachments. And what's more special, they can roll itself and make attachments. We can see how flexible the neck is and also the head is. That's very different with other fishes, cobias. This is very special of the Remoras. So that's why we do a micro CT scan of the whole Remora suckerfish. This is unpublished data. We can see that what amazed me is the Remora have a very special neck. So we can see that the neck vertebrates as embedded underneath the disc, which means that part of the disc underneath it's a neck. So that's why Remora head is so flexible. This is very unique with other fish species. So that's why we keep going and developing further highly biomimetic robots to study the Remoras. So before any of my slides, so here's our research philosophy is we build robots by incorporating its morphology, materials, sensory, and control into the robot physical body or models to implementing bio-inspired robots. And after we made the bio-inspired robots, we not only thinking about possible or promising applications, but also testing biological hypotheses to answer the interesting questions of the biomechanics, to help the scientists and the biologists to uncover some of the mysteries. So with this, I would like to thank my research lab at Beijing University. And also, if you are interested in the Remoras, you can check the our papers published in 2017 and also 2022. So thank you very much. I'm very happy to take your questions.